Great. Hello, everyone, and sorry for the delay. Thank you for your patience. My name is Madeleine, and today I'm going to be talking about my work on using environmental DNA to map the contact zone of two chorus frog lineages in southwestern Ontario. I did a background for this project last year, and I'm so excited today to be able to share some results with you all. So, as many of you will be aware, amphibians are the most threatened terrestrial vertebrate group. Understanding factors like their habitat, evolutionary history, and population dynamics can help guide the policies and strategies that will inform conservation efforts. Contact zones act as natural experiments that reveal certain elements of the ecology that can help us with their habitat, evolutionary history, and ecology, and thus guide conservation. Contact zones are areas where closely related species or lineages meet. They're common in nature and occur across a wide range of taxa. Being able to delineate the species at a contact zone and properly map where the contact zone occurs has implications for ecology. So being able to identify at a contact zone whether an organism belongs to one species or another species has implications for ecology and evolutionary history. As you can see, closely related lineages can have very similar morphological traits. So it can be very difficult at a contact zone to be able to properly delineate which species belongs to which. Some other methods to determine species at a contact zone include using behavioral traits such as calls or songs, but closely related taxa can have very similar behavioral traits as well, making it very complicated to delineate contact zones. Contact zones have particular ecology and implications for the species that are involved. At contact zones, organisms can experience a reduction in fitness. This can occur if organisms have a similar ecological niche to their heterospecifics. So in areas where they're allopatric, they are competing for similar ecological niches. Organisms can also experience a reduction in fitness at contact zones if hybrids have a lower fitness than either parental lineage. From our perspective as researchers, contact zones are also important because we want to be able to determine species range limits and accurately estimate populations. From all of these factors, understanding contact zones helps us inform conservation policy and strategy. Now, to give you an introduction to my study species, I'm studying chorus frogs, which are in the genus Pseudacris. Currently, there are 18 species recognized within this genus, and 2 lineages occur in Ontario. Pseudacris maculata, which is the boreal chorus frog, and Pseudacris triseriata, which is the western chorus frog. Pseudacris triseriata, one population has been listed as threatened by Pseudacris, and Pseudacris maculata is a candidate species for assessment. Some threats to these species include habitat destruction, fragmentation, and degradation, which can happen when roads are constructed through or between their wetlands. Urban development can destroy their habitats, industrialization, and agricultural development. Also, Pseudacris has a very unclear evolutionary history, which causes complications in the taxonomy, which makes classifying these species hard, meaning there's complications to the assessment. The evolutionary history of Pseudacris is a very contentious and sometimes confusing topic. My goal here is not to try to untangle all of these issues or explain all of the controversy, but rather just to give a bit of context that's relevant for my research. This is a cladogram based on 26 nuclear loci. What we see here is that based on the nuclear genome, chorus frogs sort into 4 clades, with Pseudacris maculata and Pseudacris triseriata in the trilling frog clade. Now, if we take a look at a gene tree based on cytochrome B, which is a mitochondrial gene, we see that the same 4 clades are well supported. However, the relative position of Pseudacris maculata and Pseudacris triseriata compared to the other species in the genus is different when we look at the mitochondrial genome. So this is just to say that looking at the nuclear genome doesn't tell us the whole story of the evolutionary history or the current population dynamics in Pseudacris. And this mitonuclear discordance is possibly due to ancient and contemporary hybridization. Recall that mitochondrial DNA is passed through maternal lineages, and so hybridization can cause a mismatch in the nuclear and the mitochondrial genomes. A specific case of hybridization is called mitochondrial introgression, where the mitochondrial DNA from one species integrates into that of another species. The extreme case of this is called mitochondrial capture, where the entire mitochondrial genome of one species completely replaces that of another species. To make the issue a little bit more complex and confusing is that currently in Ontario, all chorus frogs are listed as Pseudacris triseriata, regardless of the mitochondrial type, which is another complication for assessment. Another contentious issue, because there are many with chorus frogs, is the location of the contact zone of these two mitotypes. Previous genetic work has found that Pseudacris triseriata occurs in that southern area in southern Ontario, and Pseudacris maculata in that northern area in southern Ontario, placing the contact zone somewhere between Toronto and Georgian Bay, which is on the shores of Lake Huron. However, the exact location of the contact zone is unclear, which is where my work comes in, where I'm attempting to map the contact zone of these two mitotypes. To do this, I'm using environmental DNA, or eDNA. Organisms are constantly shedding DNA into the environment, which accumulates in the water, soil, and even the air. By taking an environmental sample such as water, I'm able to accurately represent the lineages that are present without needing to sample the organisms directly. There are many advantages to using eDNA. One that's particularly important for my study is that it facilitates the study of a wide geographic range. Catching chorus frogs takes a lot of sampling effort, and so rather than doing that, I'm able to quickly take a few liters of water, which represents the community that's present. So on the left there's a map of my sampling location, so Toronto on the southern end and Georgian Bay on the northern end. I take approximately 4 liters of water from each site and filter them using a peristaltic pump. I then collect the DNA on a membrane and extract the DNA using a chloroform isoamyl alcohol method modified from Turner et al. I'm analyzing my DNA using droplet digital PCR or ddPCR. The way that this works is ddPCR partitions the sample into tens of thousands of individual droplets, each of which is treated as a separate polymerase chain reaction. A given droplet is either positive or negative for the target DNA fragment. And I'm amplifying the DNA using a cytochrome B primer and probe set for each mitotype. So each mitotype has a different target. And this is the mitochondrial gene, which is what I'm interested in. And then I determine the concentration of DNA based on the proportion of positive to negative droplets. ddPCR is a highly sensitive technique. As you will probably imagine, DNA that's in a pond water sample is very dilute compared to a tissue sample, and so having a technique that's this sensitive is very important. Also, ddPCR performs better than quantitative PCR in the presence of compounds like algae and mud, which can inhibit polymerase activity. As you'll imagine, these are very ubiquitous in pond water. So I'm very excited to be able to share some of my results now. Based on my work, we have found evidence for a contact zone in southwestern Ontario with the yellow showing Pseudacris triseriata, the blue showing Pseudacris maculata, and the purple shows areas where both lineages are present in the same pond. It's very exciting because we have found some at mixed populations. These are just preliminary results, so I'm hoping that with further analyses, I'll be able to further refine the contact zone, but it's very promising that we have some areas where the populations are co-occurring. And I just want to highlight this work from Ying Chen, who's a co-author on my work. And she has found some admixed or co-occurring populations in the same areas that I have. So it's really promising to see that her tissue samples are substantiating my eDNA samples, and this just gives more support to the location of the contact zone in southwestern Ontario. So based on our work, we've refined the location of the contact zone. We found some distinct P. maculata and P. triseriata mitotypes, and we've also found ponds where the two mitotypes co-occur. This could have potential implications for the population delineation and assessment of Pseudacris. Like I mentioned, the mitochondrial and nuclear discordance is causing a lot of complications with assessment of these species. And so hopefully this work will help inform some of those decisions. This work also sets the stage for future studies on hybridization. Like I mentioned, we found some co-occurring populations, which is really exciting. So in the future, we are hoping to look at some genomic work for hybridization, which will have also ecological and evolutionary consequences. There are some limitations to my work. Something to always be aware of when using eDNA is that it occurs at very low frequencies in the environment. So I'm always working with low concentrations. And this is just something to be aware of when doing lab work. to be careful for contamination and everything like that, and have best practices. Also, throughout the past 2 years, we went to a number of ponds in southwestern Ontario that have historic observations of chorus frogs, but do not have current active populations. Sometimes this is because of urban development or industrialization, but sometimes the populations were just no longer there. This obviously has implications for our ability to collect our field samples, but is potentially indicative of larger issues for the persistence of these species in southwestern Ontario. This is a particular concern because in our experience, chorus frogs live in very close proximity to urban areas, and so they might be susceptible to this development and urbanization. So in summary, chorus frogs have a very complex evolutionary history. Contact zones are one element of this evolutionary history, and contact zones have particular ecology that is important for evolutionary history. Based on our work, we've refined the location of the contact zone of these two mitre types in southwest Ontario, and hopefully with further analyses, we'll be able to refine this location more. We've also found evidence for co-occurring populations, which is very exciting. This work also gives proof of concept for using eDNA for this type of survey because my eDNA samples have been substantiated by Ying's tissue samples. All taken together, this has potential implications for the classification of Pseudacris maculata and Pseudacris triseriata in southwest Ontario, which could in turn have implications for the protection that they receive under the Species at Risk Act. I want to give a huge thank you to the Lockheed Lab, the Canadian Wildlife Service, and everyone who helped with fieldwork and lab work. And thank you to the funders who made this work possible. And thank you to the conference organizers as well. And thank you all for listening.
So hi everybody. Today I will present my master's research on blanding's population genetic of blanding's turtle. So roughly I will present a context, my subject, my hypotheses and objectives, my methodology, and my preliminary results. So as most of you well know, genetic diversity is an important challenge in conservation, especially in endangered species, as we often find these species in small, fragmented, and or isolated populations. These species face a higher risk of the reduction of genetic diversity, which is important for avoiding inbreeding or having a higher adaptability for future change, for example. Genetic structure is important to take into account when we assess a population or a species, because it informs on the condition of population. For example, is there any inbreeding? Is the genetic diversity enough for viable population? It informs also on connectivity on a genetic level, but also on a landscape level that we can assess the genes. And finally, it informs on management, especially what to do and what not to do. So for my specific research, I studied the blanding's turtle, which is a semi-aquatic North American turtle that is found mostly in the US, but in Ontario, Quebec, and Nova Scotia. The Great Lakes population is considered as one large management unit with subpopulation. And the Quebec's population, the Outaouais, is considered as only one subpopulation. But it's important to note that there hasn't been any genetic study in this subpopulation before. On a geographic scale comparable to Outaouais, some studies have found that an increase of physical distance or of the landscape fragmentation is correlated to an increase of the genetic distance. But some other studies have found an inconsistent relations across the population. So for example, if we take two populations that are close in physical distance or separated by a favorable habitat, we could expect the two populations to be to have a low genetic difference. But if we take a third population that is further away or separated by a habitat that is not favorable to movement or GFO, we could expect that third population to have a higher genetic distance than the two others. And finally, if we take a fourth population, this time separated by a large body of water or by a mountain range, for example, we could expect to find this population completely isolated. So it does bring the question, what is the genetic structure of the blanding's turtle subpopulation in Outaouais? For that, I had the objective to assess the genetic diversity and connectivity, and two hypotheses. First one being that the subpopulation genetically are genetically connected to varying degrees according to topography, landscape, and physical distance. And if that is true, the subpopulation that are more physically distant will have a higher genetic distance. And my second hypothesis is that the Ottawa River is impassable barriers between Quebec and Ontario's population. And if that is true, we should observe that these two subpopulations have a great genetic distance. So unfortunately, since it's a major species that is susceptible to collect, I cannot tell you exactly where I did my work, but I can tell you that we put baited traps in 21 wetlands with 30 baited traps per wetland approximately, or 200 night traps per wetland. We did that from May and June of 2021, 22, and 22. When we caught a blanding, we would mark them with a unique ID, weight them, measure them, write them for text, amputation, any injuries, and we would take some scale for DNA samples. In 3 years, we got more than 200 blandings. We were really happy with the results. And I then processed the scales into lab, and I tested for 12 microsatellites, which range from 2 to 16 alleles per locus. So the results so far, I've successfully genotyped 198 individuals that are partitioned in 15 populations. Of those 21 wetlands, only 15 wetlands have blending turtle in. And each population ranges from either 1 to 81 individual per population. I define my population by the sites where it's sampled. So you can see here, geographically where the populations would be. It is at scale, so population 1 and 2 are closer than population 2 and 6, for example. And the 5 upper ones are from Quebec, while the one on the bottom there, population 5, is from Ontario. The first analysis I did, there is many as analysis you can do for genetic structure, but the first one I did is pairwise JOS-D. For those who are familiar with FSCs like Drader presented, it's a similar statistic, but is more appropriate to microsatellites. And the ones that have an asterisk or significantly different. Since those kind of data can be a bit hard to read, I put them in my graph. So first thing we can see is the color. For the ones that is a red solid color, it means that they are significantly different according to the pairwise JOS-D. Those in green are not significantly different. Another thing we can see is the thickness of the tree, representing the genome, estimated by those JOS. So first thing we can see, Quebec and Ontario does not seem to be different from this analysis. The physical distance and genetic distance do not seem to be correlated, which was confirmed by a mental test that was not significant. And thirdly, population 1 and population 2 seems to be different from their closest neighbors. Unfortunately, since these populations did not have a lot of individual, this I forgot to mention, but this statistics, I use only population that had 10 or more individuals in it to be relevant. So there still might not be enough individual in each population for it to be very accurate. So I pushed further and did some individual level analysis. So I took my whole 200 individual, put them in structure and did a DAPC. I'll save you the details of those things, but the overall tend to answer the question, is there any distinct genetic group with my individual? So without a priori of populations, they did take them and try to make groups out of it. Contrary to Breyer's result, here are my scatter plot, which if there were different, if there was any genetic groups, they would be separate or the circles would not be. In my case, they do not have separate clusters, which can confirm though that Ontario and Quebec do not seem to be different. So overall, with what we can take, Quebec and Ontario population not significantly different, And there is little to no genetic structure from my standpoint. But I pushed things more, I'm pushing them a little bit more. And I calculated the effective population size, which is roughly the number of individuals in a theoretical population, where they would all reproduce every generation that would have the same genetic diversity. Since genetic diversity is related to population size, we often expect that the real population number is superior to the effective population, because not every individual will successfully reproduce every generation. For some of the population, the analysis didn't work, sorry. But for 3 of them, it successfully worked, and the numbers are really, really close to the number of individual captured. Moreover, in Outaouais, the number of individual captured was 185, but the estimated number of breeders, because we don't have any juveniles, we can't compare it, is 170, which could mean that if an individual do not breed, there is a chance for genetic diversity loss every generation, which could be critical. So in conclusion, in the condition of the population I assessed, seems to be only one genetic group, few hundred adults. We did not catch any juveniles, so that the population effective size is roughly the population size of what we collected. On a connectivity level, the Ottawa River does not seem to be a barrier to gene flow, but I need to do more landscape analysis to see if there's any kind of barriers. And finally, on a management level, it is not too late since we only have one genetic group, but we might need to act. So, for example, reproduction, monetary movement tracking, etc. So, what it becomes is the landscape genetic analysis, which I will quickly present to you next. So, I hope it convinced you of the importance of assessing genetic structure in blind turtle, and a huge thanks to everybody who contributed to this interesting project. Thank you.
So we have landing turtles that extend all the way from the Ottawa River down through Kingston and so on. So I'm wondering, did you assess isolation by distance?
Yes, that was my mental test. I did not dwell on it, but just isolation by distance, like from point A to point B, it was not significant when I put the Ontario population in it. Before that, I did just Quebec, seemed to be significant, but that I had a lot of missing data, so I had to rerun my individual in my lab, and then it was not significant. So I will try to add. I have more data that I need to run after, but strictly the direct line does not seem to have a correlation. That's why I want to do landscape analysis.
Yeah, I'm really interested because it would be so lovely if you could extrapolate into Ontario so we could make some generalizations. So.
Yeah, it would be great.
Hey, hello everyone. My name is Golnar. Is it okay? Perfect. Today I'm going to talk about our projects on molecular evolution of visual genes in side neck and hidden neck turtles. Is the sound okay?
I think it's just the speaker. I think you're not doing anything.
Okay. Okay, I'm gonna talk a little bit about vision, and then I'm gonna start talking about my project. So we all know that vision play an important role in survivability and ultimate fitness of many species that prevail on Earth by providing rapid detection of predators and harmful environment. And it also plays a fundamental role in mating behaviors and recognizing prey and nutrient sources. How vision works: when light enters the eyes, it is absorbed by photoreceptor cells. Opsins, which are a very important group of protein for visions, are in the membrane of these photoreceptor cells' outer segments, and they change their structural conformation by responding to the light. Okay, so these changes actually create a visual phototransduction cascade reaching the visual cortex in the brain, which translate the visual data from external worlds inside our brain. There are two main types of photoreceptor cells in the retina, cones and rods. Cone photoreceptors are responsible for color discrimination, while rods are responsible for dim light vision. You can see cones have more diverse structure than rods. And typical vertebrate retina are duplex, meaning they both have rods and cones. There are 5 visual genes, opsin genes expressed in photoreceptor cells. The RH1 rods are the only one expressed in rod photoreceptors, which you can see in the spectral range of visual pigments that the gray dotted line, okay, that they give us the ability to see gray shades of gray in low light environments, while the LWS opsin, SWS1, SWS2, and RH2 are the opsins expressed in cone opsin gene, in cone photoreceptors, and they give us the ability to see different colors. Now, if you closely look at the different species around you, you can see that animals have optimized their visual capabilities to suit their specific ecological niches. For example, the retina of nocturnal gecko has only rods, while the diurnal gecko, very close species, has all cone retina. From our knowledge so far, we know that turtles express all 5 opsin genes in the retina that gives them tetrachromatic vision. Turtles, we all know that they are an order of reptiles consisting of 2 lineages of cryptodierans and pleurodierans, which diverged around 200 million years ago. Cryptodierans and pleurodierans have different neck retraction mechanisms that is kind of their thing and are distinguished by the way they retract their neck. As you can see, cryptodierans, they fold their neck in a vertical plane and withdraw the head and neck under the shell. While pleurodira or side neck turtles, they bend their neck sideways and tuck the head under the shell. Today, pleurodira are very not as diverse as cryptodira, and they are only limited to southern hemisphere and only freshwater habitats. Here are 2 pictures of the net retraction between cryptodira and pleurodira. So based on this study, all study that's been done on turtles, all we know about turtles' vision comes from cryptodiras, the hidden neck turtles, and we lack information on side neck turtles. So in this project, we predicted that this long divergence time, around 200 million years, between these 2 lineages resulted in considerable differences in their molecular visual system. To test this hypothesis, we first tested which opsin genes are expressed in turtles' retina in cryptodira and pleurodira. First, we reviewed visual pigment spectral tuning mechanisms, and at the end, we tested for evidence of positive selection and shifts in selective pressures between these 2 different groups of turtles. I'm going to go to each objective and report the result, and then another one. The next one. For the samples, we used 24 whole genomes from cryptodira and 4 from pleurodira, which were available on NCBI GenBank. And we had 1 eye transcriptome of pleurodira, which is really important because using that eye transcriptome, we can say which genes are expressed in the eye. This table is the result of our gene recovery. You can see that the green ones, the green circles, are the ones that were recovered fully from the start codon to the stop codon with no gaps. The orange ones are the ones with partial recovery, and the red ones are with no recovery. So we included sequences with 50% recovery and higher in our analysis, which gives us 29 RH1 and RH2, 24 LWS and SWS2, and 28 SWS1. You can see in the results of Medusa, which is a Pleurodira, the I transcriptome from Pleurodira, all 5 opsin genes were expressed in the eye of Pleurodirans, same as Cryptodirans. Here you can see variation in opsin sequences at spectral tuning sites, and the pink part at the bottom are side neck turtles or Pleurodirans, and on the top here are the hidden neck turtles. There are some slight differences between variation in tuning sites between these two, which are highlighted in in yellow. And today I just want to talk about SWS 86 and 93, which the presence of this F86, M93, and T93 in SWS1 pigment predicted to be ultraviolet sensitive. So we can say Herpetological's retina are UV sensitive, while Pleurodirans are not, or maybe less UV sensitive. For the third objective, we use PAML, which is a software package to infer direction and strength of natural selection acting on this opsin genes. How PAML works? We know that natural selection mainly operates on proteins, on amino acid sequences, and there are these changes that do not change the protein's function. These ones are called synonymous or silent substitution or mutation, and the ones that actually change the protein's function are called non-synonymous one. And using these two, we can estimate the omega ratio between synonymous, non-synonymous one and synonymous one, which when omega is equal to 1, it shows neutral selection. When it's higher than 1, it shows that gene population or that trait is under positive selection. And when it's less than 1, it's showing negative purifying selection is taking control. I specifically use random side models, M0 to test for overall selected constraint, and M2A and M8 to test for positively selected sites. We also use panel clade model to infer long-term shifts between these two distinct lineages. For the results of M0 to test for average selected constraint, you can see all the numbers are below, omega for all of them are below 1. So they are all under negative purifying selection, meaning they are more conserved. And cone opsins are under roughly twice as much constraint as rods, meaning they are more conserved than rods. And the M2A, M8 showed no evidence for gene-wide positive selection. For the clade model, we had these 29 species of turtles. We grouped them to Pleurodira as a foreground partition and Cryptodira as a background partition. This is the result. You can see omega is in between, and RH1 for the foreground, which is the field circle, the blue field circle for RH1 Pleurodirans is higher than 1. Meaning that it is positively selected. Also, but for Pleurodirans, the cone genes are under 1, which means that they are negatively selected under a negative purifying selection, and they are more conserved than rods, meaning they are more important than rods for this group. While RH1 for Cryptodirans are almost like 0, which are negatively selected, and only RH2 and LWS for Cryptodirans, the hidden neck turtles, are positively selected. In summary, we can say that opsin genes were highly conserved over these 200 million years between these two lineages. There was not much difference. There was differences, but not very much. And because cone opsins are more conserved than rhodopsins in fluoridurans. Maybe we can say fluoridurans are more dependent on color vision, while in cryptodurans, they are predicted to be more UV sensitive, which in some studies of visual ecology showed the importance of UV signaling in courtship displays and other behaviors such as nesting and hatchling. For our future direction, we are looking to expand our taxonomic and ecological sampling, specifically from fluoridurans. We are looking to find an eyeball from a fluoridurans species to estimate the spectral observance of fluoridurans and compare them with the ones from cryptodurans to see why they are not UV sensitive at all or less UV sensitive compared to cryptodurans. And to answer this question, why fluoridurans are more dependent on color vision, while cryptodurans are more UV sensitive, we need to study more the behavior of these two groups. Thank you, everyone, and I'd like to thank my supervisor and everyone who helped me in this project.
Did you notice any specific differences about terrestrial turtles? That I might have missed it there, but
no.
How were they different, or is it more phylogenetically constrained?
I tested terrestrial ones. We ran clade models to check if there is a difference in visual system of terrestrial, freshwaters, and marine ones. There was no considerable difference between terrestrial and the other two, but there was a slight difference between freshwater and the other two, which can be because freshwater the level of light in freshwater is decreasing, but like outside for terrestrials and marine, there are lots of light available in the environment, but nothing for terrestrial.
Thank you very much.
Okay. So thank you everyone for coming today. It's a real pleasure to be here. Oh no, too sensitive. Okay, so before I begin, I would just like to acknowledge that the work I'll be presenting today takes place on Treaty 7 territory. So this is the traditional ancestral territory of the Kaurna, Piikani, and Siksika nations, as well as the Ktunaxa and Tsuut'ina nations. And we honor the rights and ways of knowing of these nations and all Indigenous peoples. I've just actually moved my lab here, and so we're also on the unceded traditional territory of the Anishinaabe Algonquin Nation. Okay, so conservation, just one second, I want to get this. There we go. Okay, so conservation translocations, or the intentional movement and release of individuals for conservation purposes, are increasingly part of the recovery strategies of many species and amphibians here in Canada as well. So the numbers I have here are showing that we have about a 5% increase in the number of conservation translocation relevant species here in Canada over an earlier study that was conducted by Kelly Swan et al. In 2016. And today I want to talk about work that my lab is doing to support a specific effort to reintroduce long-toed salamanders to high elevation sites in protected areas where they've been extirpated in southwestern Alberta. And this is work that we're doing with Parks Canada, the Alberta Conservation Association, and has funding from Revive and Restore. So long-toed salamanders are one of the most widely distributed species in Western North America, as you can see here. And so you might initially be like, well, what's the concern about this species? But as you can see, this is like sort of what our field guides will show us today. The species is subdivided into multiple different subspecies, and in some jurisdictions, there's reason to be concerned about some of the populations. So most notably, down in California, we have the Santa Cruz long-toed salamander, which is a federally and state-listed endangered species in the U.S. It has a highly restricted distribution at this point in time, with as few as 24 remaining breeding locations. And when taxa get to this level of threatened, any event that sort of impacts populations can be catastrophic. So for example, in 2019, there was a trematode outbreak that caused mass mortality at 2 of the breeding ponds and put 10,000 breeding adults at risk as well. And state agencies have had to conduct sort of emergency salvage operations in years of extreme drought in California, which we know are obviously increasing, not only in Canada, but in California, but in other jurisdictions, including here in Canada and places. So I want to sort of zoom up north to southwestern Alberta, where there's still concern about the long-toed salamanders, but where the situation is considerably less dire. So this is, I'm showing a map here of the province of Alberta and the range of the long-toed salamander. In Alberta, the subspecies that is present is considered the eastern long-toed salamander, and it is listed as a species of special concern in the province because of this narrow and patchy distribution that you see. And one of the threats that the salamanders and other amphibians in the province have faced has been the sort of introduction of non-native fish into historically fish-free water bodies for recreational purposes. So this is a map that one of my graduate students compiled that shows provincial records. So these aren't even the federal records in the national parks. Provincial records of water bodies that have been stocked in the province, and you can see just how massive a problem this is. So this is obviously a challenge for our amphibian populations, but also ecologically, more broadly speaking, is a problem. So there's some interest in the province in the removal of non-native fish in some of these sites and in the restoration of wetlands to their original states more generally. And this includes interest in Waterton Lakes National Park, which is down in southwestern Alberta there, where they're considering the reintroduction of long-toed salamanders to high elevation sites where the species has been extirpated as a result of this fish stocking. And so earlier today, master's student Jana Bergman spoke about some of the lab's efforts to sort of use habitat suitability models to prioritize different sites for these reintroductions. For this talk, I want to tell you about some of the genomic data that we've been collecting to sort of help with the selection of source populations in particular. So the IUC guidelines for reintroductions and other conservation translocations make several genetic recommendations when we're planning a conservation translocation. So first, founders should show characteristics based on genetic provenance with any of the original or any remaining populations that are in the area. Second, individuals should be sourced from populations with appropriate demographic and genetic welfare. Third, founder selection should aim to provide adequate genetic diversity to the release site. And fourth, we might want to consider introducing any genetic diversity that has the potential to be adaptive in the current environment, but also into the future. And my lab is really interested in addressing these 4 sort of recommendations in this particular system. And today I'm going to tell you about some very preliminary results addressing the first 3 here. Okay, so first, with respect to genetic provenance, I want to go back to the described distributions of the salamanders again. So this is the range map that I showed you earlier. And the current map for this species would suggest that for our target area of conservation interest, which is the star down there, so that's Waterton, right? We could really source individuals from anywhere within Alberta and still be within the range of the eastern long-toed salamander as it's described. And this has implications because Parks Canada could simply go up to Banff or Jasper and pull populations from there, which from an administrative perspective is the easiest thing to do, okay? However, as a PhD student, I was really interested in using genetic data to sort of delineate the lineages in the system. And this is sort of a map of what I got at that time. So this is the same range map, but this time the shading is indicating genetic boundaries based on nuclear data. And the symbols in this plot are indicating genetic boundaries based on mitochondrial data. And so there's some indication in the system then that if you look at Alberta and our area of conservation interest there, that actually what we have in Alberta are 2 different genetic lineages. And so you can't just go up to Banff. In fact, if you go up to Banff, you'll see there's a little area where we're having a little bit of cytonuclear discordance. And so there's some indication that that's actually an area where there's some hybridization going on. Okay, so that's great, but we needed a more accurate picture or answer for our conservation partners as just to how far away from our target area of interest can we go and still recover the same genetic species and subspecies. So to answer this question, a postdoc in the lab made use of some of the samples that I had. There's also been some additional sampling that's occurred more recently. And we included in this first analysis 53 representative samples. We were interested in using sort of genome-wide variation. So we use DD-RAD, or reduced representation sequencing. Many of you will be familiar with it. And depending on how the filtering is done and which samples are included, we're getting about 35,000 SNPs. And so this is from samples that are across all of the different lineages, but with a little bit more of a concentration in southwestern Alberta and southeastern BC. And so here's a first look at what the genomic data are showing in terms of lineage boundaries. So in this particular plot, the colors aren't that informative. This is a PCA with just axes 1 and 2 shown. So we're looking in genetic space here. But I want to walk you through how these different populations that we have, each dot's a population, how they're falling out in this genetic space. So the groups that I have circled in red actually are individuals from the more western subspecies. So we're looking at California, Washington, and Oregon. The groups that I've got circled in green there are populations from northern Alberta, so including Jasper, Banff, and into BC for the other Rocky Mountain National Parks. And then the group in yellow is individuals that are from southwestern Alberta, southeastern BC, and south into Montana. And that includes over here our populations from Waterton Lakes National Park. And I'll just note this sort of middle grouping here. These are individuals from Banff and the Kananaskis area. So they're kind of falling intermediate, as we might have expected, for the yellow lineage. I'm going to start calling them yellow and green, but they're intermediate to the yellow and green in the individuals in the province. And just interestingly enough, I want to note that the sort of boundaries that we're seeing, at least between the yellow and green, are kind of falling out. There appears to be this real gap in the species distribution in the province. So I've got in that red box the Highwood watershed encircled, and this is an area where Larry Powell and others back in the day said we haven't found salamanders here, but we've gone back and done extensive surveys and we do not find salamanders there. So that's kind of interesting to us because the group that's circled in yellow are falling out south of that gap. The individuals that are sort of admixed are falling out right north of that gap. So from the perspective of, the translocation, we've got this picture that the park can go as far as the Highwood watershed, but no further if they want to sort of stay within the yellow lineage. But from a phylogenetic perspective, this is also really interesting for us. What I think we have, what historically was a sort of arm of the yellow lineage moving up through eastern BC and entering Alberta, sort of north of that gap there, and intermixing with individuals from the green lineage, and that we have something quite different in southwestern Alberta. And we have the data to test that, so stay tuned. That's a different story, but this is what the picture is looking like for the conservation translocation. Okay, so our results so far have clarified where the boundaries between the different groups are and how far the park can go to sort of source individuals. The next questions that we had were about the genetic health of the populations, and here we specifically wanted to avoid removing individuals from existing populations that show a genomic signature of being vulnerable themselves. And we also want to make sure we're not, populating our new release sites with highly inbred individuals. So towards this goal, we went and did some finer sampling of 45 sites in Waterton and the surrounding area. And we're looking at 5 to 10 individuals per site, and we're using them to estimate some of our common populations genomic metrics. So here I'm just going to show you some very preliminary results that were actually included in a master's student's thesis in the lab. And so I'm showing you allelic richness and expected heterozygosity. And you can see, all I want to point out is that the populations really differ with respect to the levels of genetic diversity. Okay, and so that suggests that that's kind of giving us an initial insight on how we might prioritize different sites for release, but we have many more sites to look at. So to summarize, our work with the long-toed salamander demonstrates the potential use of genomic data for clarifying the boundaries between distinct groups. Specifically, we've shown that long-toed salamanders in Alberta belong to 2 groups with a zone of admixture in the Banff region. And in terms of the conservation translocation, the park would be best to source populations from south of the Highwood watershed if they were interested in maintaining genetic provenance. Second, we've been using the data to assess the relative genetic health of potential source populations. And we've shown that the sites have different levels of genetic diversity. And so it might be interesting for the park to source individuals from outside of the park or to consider sourcing individuals from multiple sites, depending on which populations they want to pull from. And so we have a lot of additional analysis underway. I'm just really going to quickly tell you what we've got coming up next. In general, I think for the conservation translocation literature, we've got some big questions like how distinct do genetic groups need to be before we even worry about this? Are there thresholds for genetic diversity that we want to consider before we remove individuals from populations? And then, of course, in our work, we need to have some empirical tests of the value or the role of genomic data in contributing to translocation success. And I want to point out here that with something like the long-toed salamander, we're not down to just one population. We can fiddle around and maybe do some sort of cool, bigger experiment looking at whether those source populations matter. We have lots of data, many tissue samples. So if anybody's interested in this species in particular, these types of questions, please come talk. I'm very interested in meeting collaborators and potential students. I'm done. Sorry, Nick.
Thank you very much for the presentation. That was great. I like to see the genetic analysis of observation. It's completely lacking. I have a quick question, especially when it comes to this species.
Yeah.
They have an extreme site, a breeding site fidelity. So how is that being considered in conservation?
I think we need more data on that. It depends. I mean, we see these salamanders roaming off in places that we're surprised that there's one population in the park in particular that we are pretty sure is a recruitment source. It's actually been targeted for a road mitigation effort, and they've basically built a $2.5 million road to funnel the salamanders into the sink population where they get eaten by fish. And so there's a little bit of work to be done to sort of assess that, because clearly those individuals which are still there, well, the same individuals might not be there, but that we still see the salamanders using that. So if that's a dead end, they must be going somewhere else. So I think there's some more dispersal than we think.
So hello everyone, my name is Ying. I'm a PhD student at Dr. Stephen Lockey's lab, and I will present our work to generate a reference genome for Western forest frog. So as we are all aware of, we are in a mass extinction, according to the report from Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. Around 1 million animal and plant species are now threatened with extinction. And among those, amphibians are one of the most threatened group, with over 40% facing risks of extinction. And the good news is that we now have many tools to conserve species. Some examples here include population genomics that will identify genetically distinct populations for management, as we have heard a few talks, and we'll hear a few more. Environmental DNA tools or CRISPR tools to detect species and biodiversity across landscape, as has mentioned by Madeleine studies. And some reproductive technologies include cryopreservation of cloning and biosynthesis tools that can be used to help wildlife to fight diseases. And another super powerful genomic tool is having a contiguous, accurate, and annotated reference genome. So yeah, genome is a blueprint for all species, and it tells us how different genomic elements arrange spatially, where the functional and non-functional elements locate and interact, and which genomic regions differ among populations and species. And those information can be critical to understand species divergence and adaptation, to infer population demography and viability, as well as predict evolutionary potential under climate change. And so a genome serves as a powerful tool where primer designs and other genomic applications can achieve. However, generating a high-quality reference genome is a non-trivial task, and it's only in recent years that's possible for non-model species. We have over 7,000 frog and toad species, according to FBA Web. And every year, new species are described, often with genetic evidence. In contrast to the species diversity, the genomic resources are relatively limited compared to other taxa, such as mammals and birds. So far, we only have 44 genomes assembled, which is only 0.5% of the species diversity. And half of them are county and scaffold level, which just means the genome is fragmented and gives much less power in showing the structural variants and so on. So the best genome will be a complete genome, which means all the chromosomes are complete with no gaps, with no unlocalized and unplaced scaffolds. So that species is the mountain yellow-leg frog. So we are interested in Western forest frog, which is a species of conservation concern, as Madeleine and Jeffrey mentioned before. The Great Lake population shown in gray on the map are listed as threatened. And there's debates on the taxonomy. Some authorities have already listed the Great Lake populations as boreal forest frog based on the mitochondrial DNA. While the nuclear DNA show that the Great Lake populations are more closely related to the Carolina population shown as red than to the boreal forest frog from Western Canada. So to facilitate conservation and understand evolutionary history, we are working to generate a reference genome. We have collected a male individual from the Clearville Conservation Area near Toronto, and that individual has the Western forest frog mitotype. And this field work is under the help from John Britt and Jennifer Thompson from Canada Wildlife Service. And all the tissues have been stored in RNA later in -80. And we have obtained PacBio Reveal hi-fi long reads and high-C contact map data, which are the state-of-the-art technology to generate a chromosome-level reference genome. We are following the Bird Genome Project pipeline by using hi-fiism to assemble and place the genome, following with the manual curation to correct scaffolding errors and false duplications and so on. So this analysis are currently ongoing, and we aim to publish the genome this December. So stay tuned. And once complete, this will be the first genome in subfamily Acrocinin, which includes chorus frogs and cricket frogs. And this genome will greatly aid conservation genetic study, as well as give us much more power to investigate the evolution history. So I would like to thank the funding from NSERC and Digital Research Alliance of Canada. And I would like to thank all the help from the people listed here for field work and lab work. And that's all. Thank you.
All right, thanks so much. As mentioned, my name is Christine Drader. It's my first year at CHS, and I've been with the Adopt Fawn program since April of 2021. So very excited to be sharing today about our milk snake field project and Ophidiomycosis. So Ophidiomycosis, it's a fungal disease by the fungus Ophidiomyces ophidiacola. It's documented in over 49 native snake species in North America, and clinical signs include dermal lesions such as scabs, crusts on scales, underlying pustules or ulcers, facial swelling, abnormal molting, or white, opaque, and or cloudiness of the eyes. So our study site is within the Rouge National Urban Park. It's outside of the Toronto Zoo, as some of you may know. And we've been doing artificial cover object surveys since 2011, excluding 2016 and 17. Our target species is the eastern milk snake, but we do monitor any other snake populations we come across. So the primary goal of this is to identify their presence within RNUP, monitor their long-term population trends, and identify their kind of seasonal habitat use in hibernaculum sites. So when we find snakes under cover objects or opportunistically, we process them, and we do record notes on and swabbing for Ophidiomyces signs. So throughout our surveying period, we've observed a number of snakes. However, routine testing was not done until 2020. Prior to that, only one observation made each. It was kind of extreme cases in 2012, 14, and 18. But then since then, we have seen an increase of signs after we've been continuously swabbing. So just looking at 2020 and onwards, when signs were come across and observed, infected tissues specifically were swabbed, and then the sample was frozen and then later sent to the Canadian Wildlife Health Cooperative for PCR testing. In 2020, we had submitted only 5 of these samples and 1 came back inconclusive. In 2021, we submitted 17 samples and 1 came back positive. And then last year in 2022, we sent 18 samples and 7 came back positive. And then it was noted that in those results that it can't be completely ruled out as negative because there can be more issues with skin swabs and false negatives. So due to this increase of positive samples, we spoke to Dr. Christina Davy and we decided that we'd like to try to identify the full extent of Ophidiomycosis in the environment. So this year we modified our swabbing results. So in May and June specifically, we swabbed all snakes, regardless if they presented signs or not. So we swabbed the entire length of their body. And then if they did have signs, we focused on those tissues. But after that, in July till the end of our surveying period, we're going to be swabbing only snakes showing signs for funding purposes. And then prior to 2023, I just want to note that it was only the tissues sampled, but here we're still swabbing the entire length, but with focus on these any sort of tissues that are showing. So the results this year, we swabbed 44 snakes. 27 had signs, 17 without. Of the 27, 12 came back positive, 2 inconclusive. So again, we're seeing slight increase from last year. And then 17 signs, they all came back negative. So not much to be seen there. And then just kind of to bring up species-specific prevalence, despite observations, we see eastern garter snakes, northern red-bellied snakes, and Decay's brown snakes. But signs of infection are primarily observed in our eastern milk snakes, and it's solely confirmed. So only positive cases have been seen in our eastern milk snake population. So although we do catch more eastern milk snakes, this proportion of seeing signs is in our higher in our eastern milk snake population. I'd also like to highlight a very interesting case, personally to me. This is Chocolate. She's first captured in 2013. She has a missing right eye and a stubby tail, but eastern milk snakes can also be identified through their head pattern. And in 2014, she had a biopsy done in-house by the Toronto Zoo since she was noticed with lesions. And this came back as a fungal infection of the Fusarium species. So this is considered at the time non-pathogenic to animals, but the large number of organisms within this suggested a disease process. So after this, after our 2020 introduction to swabbing in 2021 and 2022, she was tested for Ophidiomycosis, but both of those came back negative. So perhaps going forward, we're going to consider testing for this Fusarium species in Chocolate, as well as our other snake fungal swabs that we get. Lastly, Christina Davy is setting up a cross-Canada study of Ophidiomycosis and is recruiting collaborators for 2024. So please feel free to take photos, fill out that QR code, or ask her all about that project. And then I can flip back if necessary, but acknowledging all the partners and individuals that have been involved in our project, including many current and past members of Adoptapod. Thank you very much.